All right, everybody say hi.
Hello.
See, that's important. And just like the clacker. You can hear yourself. So, if you were watching, we were starting to go through our macromolecules and building up our chart of the basic macromolecules and incorporating the idea of bond strength with that. And so we talked about carbohydrates, and their main claim to fame is that we can build really long glucose polymers. There are a lot of other sugars that are used for polymers, but starch and cellulose people are fairly familiar with. This is a little throw out or throw up. Things like gelatin is another glucose polymer that's also got a little bit of protein in it. It's what's called a proteoglycan. Lots of proteins are glycoproteins. They have a fair amount of sugar on them, but still a pretty big percentage of protein. Pectin is another carbohydrate polymer that plants produce. If you peel an orange, the white part underneath, that's pectin. And it's used as a gelling agent. Anybody ever make orange marmalade or jelly of any kind? The whole point was we were talking about carbohydrates. You heat them up. These long chains are all balled together and held together by hydrogen bonds, the intramolecular hydrogen bonds. You heat it up and whoosh, they open up. And then when they cool, they form intermolecular hydrogen bonds, these real long strands all hooked together, making a gel. So we've got all these different kinds of gels.
You said beforehand they were intra or inter?
Inter is within. So when they're a little ball, the hydrogen bonds are within the molecule. So I think of it as you have the interstate highway system, but nobody talks about an intrastate highway system. So the ball might look like this, where we've got hydrogen bonding holding this ball together. And so you've got little ball bearings of starch, and then only when you heat it up does it whoosh open up. That's with heat. And then when it cools, you have these long strands now that are opened up, and they form hydrogen bonds between individual molecules, and so that would be intermolecular hydrogen bonding. So carbohydrates then serve, obviously as a good calorie source. We'll be burning them and generating ATP as we get into metabolism. But they also are a really good source of cushioning and a material that can hold a lot of water. And they also have a very strong negative charge. And so the outside of a lot of cells have a lot of sugar and glycosylations on them. The next thing we went into was our discussion of the lipid polymer. And lipids and fats don't really hit the polymer model quite as well. The monomer is the fatty acid. And we threw out the idea of the 18-carbon fatty acid as a really standard building block. And if we have it completely saturated, that is, every carbon has got two hydrogens, that's stearic acid. It's a bit of a wax. And then you've heard of things like perhaps omega-3s and omega-6. And it's one of these things probably every semester I look up in Wikipedia after this lecture and go, oh, right, exactly where the double bond is. It's 3 carbons in or 6 carbons in. And the main point here is that we've got double bonds, and I'm not going to draw the entire fatty acid out. The hydrogens are fixed in space relative to each other because of the double bonds, and consequently you have an orientation that is going to be fixed. This is the trans configuration, which in fact is not found in naturally occurring fats. We've got always the cis configuration, cis same side. And what a double bond does is it makes the fatty acid actually have a kink to it. Like that. So the presence of double bonds means that the fats can't pack together quite as well or as tightly. And so the presence of double bonds means you're going to have a liquid oil at room temperature. It lowers the melting point. And this is an important thing because membranes are made up of fatty acid tails, as we're about to get into. And on a cold day like this, and in fact as winter comes on, all our little bug friends and the trees, they actually have to modify their lipids in their membranes to keep them from freezing up. That'd be a very bad thing if you were like a little insect or even a lizard. For your membranes to all seize up, you got to keep yourself fluid. And so there actually is a change in the double bond concentration as the seasons begin to change. So you can really freak out animals and plants by taking a wintertime guy and raising the temperature up, to 75 degrees. They will have a little bit of a wobbly time for a while. But the thing that makes the fatty acid class so interesting, besides forming the cream filling in Twinkies and Bean Buns, is this. We can begin to make kind of a polymer. What we're going to do is take a glycerin molecule, which is a 3-carbon, what's known in the biz as a sugar alcohol. It's 3 carbons with 3 OH groups coming off, and the rest is hydrogen. This is glycerin, or glycerol, and you perhaps know this. It's a humectant. It's something that maintains moisture in a lot of baked goods. It's vaguely sweet. It's found a lot in cosmetics. It's probably found in some hand sanitizers. It gives it a little bit of thickness and gooey rather than just being alcohol in your hands. And this is what we're going to start hanging fatty acids off of. And this is a really good example when you're not sure how biomolecules are going to link together in terms of making a polymer. Always look to see where you can make a molecule of water. So, we're going to take a fatty acid and the carboxylic acid part of it is here, and there's the long tail. What we're going to do is do a dehydration or a condensation, depending on your perspective, reaction, and meaning that we're going to make a molecule of water. So let's take that hydrogen, and that oxygen, and whoop, we squirt out a molecule of water, and we have linked through this oxygen the fatty acid. All right, so this is something that's called a monoglyceride. And they're definitely floating around in us. Plants have got a lot of monoglycerides and things. Olive oil is a mix of mono and di and tri, which I'm about to get to. If you look on some of your fancier baked goods or more highly processed baked goods, they will add monoglycerides, and I'm sure Archer-Daniels Midland will be happy to sell you a freight car load of monoglyceride. What they're doing is distilling out these different fractions from soybeans or whatever. So that's a monoglyceride. The next thing you could do is add another fatty acid, and of course this is a diglyceride. And now we're going to have, can you guess what this one's called? You're right. This is a triglyceride. And I'll just tell you, you probably are not yet fortunate enough to be old enough to have a full lipid panel on your blood when you go to the doctor yet, but wait another 10 years and they'll do this. And one of the numbers they give you is your triglyceride number. And your triglycerides represent the amount of this fatty acid polymer floating around your bloodstream. And so we have a lot of fats that are being transported around, probably incorporated up in some kind of protein and things. But anyway, this is a triglyceride. So you can get in the food industry that you can purchase mono, di, and triglycerides and combine them, and that's probably what makes things like industrial baked, cinnamon rolls, have that perfect gooey texture that you can't quite get at home, and it's because you don't have access to this sort of stuff. All right, so when you think of blubber and lard, this is what you're looking at, pretty much all triglyceride. And we can have different double bond combinations in here. It could be all saturated fat, and that's going to be like what lard is, so it's like, solid at room temperature. You can throw in a couple of double bonds in different spots, and you can cause these guys to kind of kink out and spread apart, and then it'll be more liquid. And so this is what a lot of corn oils and liquid oils are. So, okay. Now, what makes these guys a really important class, though, is the following. We can take one of the fatty acids off. So we have a diglyceride. and now add a phosphate group. And while you weren't here for that, you do know that this is one of the critical things that you should use either a tattoo or Sharpie to etch into your forehead, leave some space for other things. But that is that the phosphate group carries what? A massive negative charge. Thank you. So, whenever we see phosphates, we're always thinking massive negative charge, or just negative charge. And now we're going to actually swing this guy up, so it's kind of like not interfering with these nonpolar heads. And generally, we add some kind of decorating charged molecule. It can be a sugar, like the sugar inositol. A standard decorating molecule is a nitrogen-containing molecule called choline. This is extremely common. And when we flip this around, we end up now with a molecule that's really quite schizophrenic. It's got a negative charge up here because of the phosphate and the choline, which actually has a positive charge on it, but regardless, it's still got a really strong charge to it. And then we got the two fatty acid tails. So just imagine spinning this phosphate up, and that's what we're drawing. Now, in the typical sort of diagrams, what we do is you draw the polar head and then the two fatty acid tails. And this molecule is said to be amphipathic, meaning that it's got a charged end. It is very polar, and it's got a nonpolar tail. And if you want an example of this, crack an egg. Egg yolk is almost entirely phospholipid. And in fact, with choline on here, this molecule is known as phosphatidyl choline. For whatever reason, in the food biz, this same molecule is called lecithin. So next time you're eating chocolate, next time you open up a jar of mayonnaise, pretty much any kind of moderately processed food, you'll see lecithin as one of the ingredients. And I'll tell you why. First off, this is the key behind forming the phospholipid bilayer. And it works really well because if we take like a red blood cell, just as an example, the red blood cell is floating in serum. And that is primarily a watery aqueous solution, actually with a fair amount of ions. And the cytoplasm is also aqueous with lots of organic acids and lots of ions and things. And this allows the phospholipids to sort of operate like this, where we have the charged heads facing out, and we form then the lipid bilayer. And this is able to float around, and by putting in double bonds, we can make it float around even more. Yeah. All right, so now a couple of things just interested in terms, not so much natural systems, but in biochemistry. We use phospholipids and amphipathic molecules a lot because this is what a detergent is. Regardless, if you take a phospholipid and throw it in the water, it'll form something that'll look like this, where the fatty acid tails are hanging out, having their own little nonpolar party, and the phospholipid heads are hanging out with the water. So we got water out here, and this is a stable molecule. Now, let's sort of think about the effect that a phospholipid has. If you were to take a jug of olive oil and put a couple of glugs into water and or vinegar, making a nice oil and vinegar salad dressing, and you shake it up and let it sit on the counter for a few minutes, what's going to happen? The oil is going to separate out, and the vinegar or the water will be below it. So this is an example now, kind of a polar, non-polar, self-organizing thingamabob, in that the polar molecules are not going to be miscible or able to interact with the non-polar molecules. All right. But if you add phospholipid, something magical occurs. Because I just started thinking about this as an example. I personally don't like eggs so much. I mean, I put them in things. I'll eat an omelet once in a while. But making scrambled eggs, the white is pretty much all protein and water. The yolk is all phospholipid, almost all phosphatidylcholine. It is the membrane to be for the little baby chick. And if you take your whisk that up, you make a homogeneous yellow mix, right? Now, we don't advise this, but if you let this sit on the counter for 15, 20 minutes, ain't nothing going to happen to it. It's going to stay a homogeneous yellow color. And that's because the water and phosphatidylcholine are now actually interacting in a way. This little structure, this little blob here, a bubble, is often known as a micelle. Now, a really cute trick you can do, micelle, is you can begin adding straight oil to a solution of phospholipid. And the oil then is able to get trapped in here. And we have made a stable mix of oil and water. This, of course, is what we know in the business as an emulsion. Consequently, phospholipids, and it is true for all amphipathic molecules, they are emulsifiers. And one of my favorite emulsions is mayonnaise. I'm not sure how many S's there are in mayonnaise, but today there's 2. How many of you are anti-mayonnaisers? Oh, we have a few in the audience. You at home, think about it. Really, I love mayonnaise. And actually, most recently, a very old now, but wonderful chef, a guy named Jacques Pepin, who does little videos from his home. He used to be the chef for Charles de Gaulle, who was the Prime Minister of France after World War II. And he and Julia Child, who maybe you don't know her either, regardless, he was making a mayonnaise. And all you do is take an egg yolk and you mix in like a cup of oil, and you just start whisking it. And it used to be that you had to, whisk, or the idea would be you have to whisk very slowly, and you have to take a lot of time. He just was taking his little whisk and making mayonnaise. It's not quite as creamy as your Hellmann's, but it's actually quite amazing. And so, if you like mayonnaise, you should try it. If you don't like mayonnaise, I don't know what to tell you. This kind of emulsifier, though, this is found in all of our sort of Thousand Island dressing things, and there are other molecules that you can use as an emulsifier. But any time that you're like at a cafeteria or stuff, and you have like a Italian salad dressing that's in one of those great big tubes, or tubs, and you can dunk the ladle into it, it's got an emulsifier in there to keep the oil and the vinegar solutions mixed together. Otherwise, it'd separate out and it wouldn't work very well. So, there you go. All right.
So, like, the definition of emulsion is like?
Well, an emulsion is a stable mix of lipid and nonpolar solution. A stable mix of oil and water. And just thinking here, pretty much in terms of our biochemistry, we don't really form emulsions so much. What we do is have cell membranes that can do that, but that only happens if you lyse and stuff. Anyway, so the next segue, and this is again sort of a tool of biochemistry, is all detergents are emulsifiers. And our favorite detergent in the lab, one that we use all the time, is a molecule known as sodium dodecyl sulfate, otherwise known in the biz as SDS. And at the end of the semester, we'll be doing some SDS gels. Sodium dodecyl sulfate is an amphipathic molecule. It's got 12 carbons, that's a dodecyl, and it has a sulfate, which you probably should, and when I say sulfate, you don't want to jump up and yell negative charge, but you should definitely think negative charge. Sulfate's got a moderate negative charge, not quite as good as phosphate. So you got this sulfate head, and then a 12-carbon tail. And that makes for an amphipathic molecule. And if you think about the idea of detergents, what they're doing is they are actually emulsifying oils. And so if you've ever experienced, working with something that's really dusty, drywall dust, you can suck all the oil out of your skin and then things don't stick. But if you put your hands into something oily, then you got all this dirt sticking to you and it's quite a mess. Our skin has a fair amount of oils that we secrete, and dirt and bacteria and viruses can get trapped up in that oil. So in order to remove the oil, if you just wash your hands under water, you can't strip the oil away. But if you add an emulsifier, a soap, a detergent, mix it up, and now all the oils get washed away. So that's one of the things that we deal with when we're showering and using different kinds of soaps. And if you look on your labels of toothpaste, any kind of detergent, soap, toothpastes actually have to list things by name. As you'll see, sodium lauryl sulfate is the term for 12-carbon. And that's a really good marker. Ha! So, yes? What's the odds? Ah! I'm taking a valuable video time, but I'm having a ball. Is this gonna? Let's see. Ah! So it is known as sodium lauryl sulfate. That's the whole game with organic chemistry. Is everybody's got at least two names. You have your Facebook name, you got your email name. It's a way of keeping the riffraff at bay, so you never know what the molecule is. An acetate, an ethyl group, whatever. Sodium lauryl sulfate, it's the same thing as sodium dodecyl sulfate. And pretty much all the soaps you use are sodium lauryl sulfate. Now, if you're into shishi soaps, they may be, lauryl sulfate free. Oh, well, it'll be sodium laureth sulfate, which will have a double bond in it. And it's lauryl sulfate free, but it's the same thing. The more amphipathic the molecule is, that is, having a really big charged head and a really big nonpolar tail, the more harsh the detergent is. And sodium lauryl sulfate, it's a pretty strong detergent. So you got your Dove, it's 1/4 cleansing cream, which is essentially sodium lauryl sulfate. They just now throw oil into it, so some of the soap is actually kind of taken up with the oil in there. Just use less soap. I don't know. Baby shampoos are another great thing in that sodium lauryl sulfate has been viewed as being too harsh for your baby's delicate skin. So you find all these lauryl sulfate-free shampoos. And no matter what, if it's going to be a soap, it's going to have an amphipathic molecule. That said, this is called an ionic detergent because it's got the real strong negative charge. There are things like woolite or what are known as non-ionic detergents. And these are alcohol polymers, and really what they are is just not very good soap. Okay, so we use detergents in the Molec and Cell lab all the time because when we have to disrupt a cell, you have to lyse a cell, you're going to have the phospholipid bilayer, you're going to have lipids and things, and you need to solubilize all those guys. Additionally, when we start getting into proteins, SDS can help to solubilize proteins that would have a really big nonpolar stretch, and it'll solubilize these things. And so we use SDS to isolate proteins, to lyse cells all the time. It's a pretty harsh detergent though. So that is what we had to say about the fatty acid class of molecules. They are typically then nonpolar. We can adjust the melting point of them by changing double bonds. They are obviously a really good source of energy. We'll get into that a little bit later. And there is the phospholipid class, which is not fitting into the nonpolar part, but phospholipids and detergents being amphipathic, they interact with water and nonpolar molecules in a really interesting way. And the last thing we'll throw in here, just because if we don't hit it here, I don't know where we hit it. Not all of our lipid class are fatty acids, but that's the way you go at it in terms of if somebody asks you, look out. There is another molecule in the lipid class that we would be remiss, and that is the sterol, which is the base for making steroid hormones. And its base is typically cholesterol. Additionally, vitamin A and vitamin D are based on the cholesterol structure. Cholesterol is a rather complicated heterocyclic molecule, but it is my pleasure to diagram it for you. It's something like this. I don't know what the hell cholesterol looks like. And I will tell you, if you bother to memorize the structure of cholesterol ever in your life, you're wasting your time. Really? Why? Cholesterol is a heterocyclic molecule. Look it up in the book. What cholesterol can do actually is it gets placed into membranes, particularly in animal cells. Plants have different kinds of sterols that have cholesterol. And it serves as a stiffener for the membrane. So you can actually embed cholesterol into the plasma membrane and sort of stiffen it up a little bit. So then we have steroid hormones, which when we start talking about hormone mechanics, just remember steroid hormones are based on cholesterol. They're very nonpolar. Thank you very much. All right, so that was our fat class. We've got phospholipids. We can make cream filling. We can make squishy breads. We're rolling. Now, the other major food class. Ooh, my coffee is hot today, but that's good. The other major biomolecule class that is next, and arguably the most important, sure, and that is the protein class of macromolecules. So you're building your chart of our biopolymers. You got carbohydrates, sugar polymers. You got fatty acids or lipids, fatty acid polymers, and we can make phospholipid. Now we come to proteins. Proteins, the monomer polymer model works really well here. The base monomer is the amino acid. And for pretty much our purposes, there are 20 naturally occurring amino acids. So, in terms of actually seriously structure, I don't say that much about drawing structures, but this is a structure you probably should know. And that is the structure of a base amino acid. If you're going to take biochemistry, you'll have to memorize the individual 20 amino acids, start now, whatever. This is the base structure of an amino acid. It's got a carboxylic acid tail, it's got a central carbon, and then it has an amine group, that nitrogen-containing amine group. Depending on the pH, you can have 2 or 3 hydrogens, whatever. This central carbon has got a hydrogen on it, and then it's got what is known in the biz as the R group. The R group is one of 20 different side chains. Now, what makes amino acids really sing is if we consider the previous 2 biopolymers, there wasn't a lot of differences in the molecules. I mean, just as an example, starch or amylose or glycogen or cellulose, these are just glucose polymers. As far as the eye can see, it is just glucose. It's all got a strong negative charge. It forms these hydrogen bonds, yes, but there's not a lot of charge heterogeneity in there. All right, with fatty acids, at least in terms of triglycerides and fatty acids, it is just a long nonpolar tail. It's just a very nonpolar molecule. And yes, the amphipathic part can form up some structures, but it's still a fairly heterogeneous molecule. We now have the opportunity to make some really complex molecules because these 20 different R groups have got distinct different characteristics to them. And there are a number of ways you can kind of lay them out, but here what we're thinking about is that the R groups can be polar, they can be negatively charged or positively charged, they can be acidic, they can be basic, they can be nonpolar. So we now have an opportunity for a lot of different R groups and lots of different cool interactions. Now, my favorite R group is the hydrogen. And this is the amino acid known as glycine. So when you're memorizing your amino acids, you always know glycine because it's the hydrogen. So you only have 19 more to memorize. Alanine, I think, is a methyl group. That's an easy one, too. So some of them, like tyrosine, end up actually having a phenol ring in them. Others have multiple 4 or 5 carbons coming off. Phenylalanine actually has some weird ring structure and little kinks and things. So the different amino acids have different kinds of structures. And let me just see here. I'm looking at panel 27. Not at all do you need to worry about this. Oh yeah, at least in my book, this is volume, whatever, 4th edition, page 75 has got all the, and 74 and 75 has got the basic side chains, acidic side chains, uncharged polar side chains, nonpolar side chains. Memorize them up? No, not for me. But while we're here, let's go ahead and link up to amino acids. Let's make the beginnings of a protein, an amino acid polymer, a polypeptide, all these terms. What we're going to do here is look to see how we can make a molecule of water. We're going to take the OH group off here. We're going to take the hydrogen off here. And bada bing, we make a carbon-nitrogen bond. It's just a plain old carbon-nitrogen covalent bond, one of our strong ones, but it is given the fancy name of the peptide bond. Now, another critical part behind thinking about proteins is they are built from the starting end back. And we have an orientation now to these molecules such that, just looking for a different color here, we've got on this end, this is referred to as the amino terminus, and this is referred to as the carboxy terminus. And there's a relationship as well when you're looking at an RNA. Remember that with genetics, and we'll be getting into this in a lot more detail. This is going to be equivalent to the 5' end. This is the 3' end. We've got an orientation now. And if you're the first amino acid, you're line leader, and everybody else has to farm up behind you. You always are adding new amino acids to the carboxy end. So we're going to just start adding on more amino acids. So we always are going from the amino terminus and adding to the carboxy terminus. So when we begin biosynthesizing proteins, you're always adding at the carboxy end. Now, this is a dipeptide, not all that exciting. We have a range of different kinds of protein-based molecules floating around in your body and in different cells. We've got, individual amino acids are being used as neurotransmitters. We have dipeptides that are being used also mostly as neurotransmitters. We got 3 and 4 amino acids linked together, tripeptides. Some of these are used as signaling molecules. Insulin, probably you've heard of insulin. Insulin is actually a rather modest protein. Insulin is 50 amino acids. That's insulin. One of the main proteins in muscle is a protein called myosin, and it's got somewhere in the range of 500, 700 amino acids. I've never bothered to count. The point being that you can have very small proteins, or called peptides, up to really great big amino acid polymers. Big ass proteins these are. Now, what really makes this whole game start to jump is the fact that these R groups can begin interacting. And we've got these 20 different R groups. So, let us now look at how that lays out. And what we're going to get into here is, in fact, truly the answer to life, the universe, and everything. I mean, this is, in fact, the fundamental mechanism behind mutagenesis. I'm getting so excited that I actually have to take my sweater off and roll up my sleeves. On Monday's lecture, I just didn't, but today, I am fired up for proteins. All right, so let us imagine we've got an amino acid chain. And let's now draw it out. I'm getting ready here. Hold on. God, having myself videotaped for posterity is embarrassing. But, hi everybody. All right. So rather than drawing out the actual structures, you could, but we're not going to. We're going to draw it out like this, where we got the central carbon, the peptide bond. There. Now, let's put some R groups on here. What we're going to do is have a stretch of negatively charged R groups over here. We'll have a stretch of positively charged R groups here. And we'll have a stretch of nonpolar R groups here. And let's see what happens. What we're going to generate is a level of structure, multiple levels of structure. And this is, again, a really key point to this whole biochemistry biz. All right, so we've got what's called the primary structure, and just for abbreviation is a 1, or the primary term is symbolized with a degree. We have the primary structure. The primary structure is defined as simply the amino acid sequence. And this is held together by the peptide bond. So it's held together by the covalent bond. Now, in our bond strength chart, we know the only way to break that covalent bond is to set it on fire. We can invoke, and in lab we will, some enzymatic wizardry where we can actually use enzymes to break the peptide bond, proteases. But for now, we're not talking about that. We're just thinking about the kinds of things you might be able to do in a cell standard environment. And the thing you can't do is break covalent bonds. So this is a tough-ass bond. It ain't going nowhere. The primary structure is really not going to be subject to degradation. But the next level is what we call, not surprisingly, the secondary structure. Now, secondary structure is defined as the neighboring R group interactions. And neighboring means who are the R groups within maybe 5 amino acids of you on either side. Who are the R groups in your neighborhood? And the R groups now begin to interact with each other. And let's just think about this for a second here. We got a stretch of negative charges. We know like charges repel each other. And so these negative charges, they're kind of beating on each other and poking each other in the ear. And mom calls out, if you boys don't stop that, I'm going to turn around, and what we do is I'm going to flip every other one of you, so you're going to be sitting down on the bottom of the floor, and you're going to be sitting up in the chair, and you can't be poking your brother in the ear. I had 4 brothers, and my mom used to do this to us. No, but what we do is we spin the central carbon such that we're going to alternate the sides of these R groups. And we're going to do the same thing over here. And that spinning actually induces a spirally twist to the protein. So we're going to have still our nonpolar guys here, and then we're going to have another spirally twist here. And this spiral twist is known as the alpha helix. Now, another standard secondary structure, a little bit harder to draw, is when every fifth or so amino acid has the ability to hydrogen bond with the dude 5 amino acids behind it, you can form a hydrogen bonded zigzag. This is known as the beta pleated sheet. And these secondary structures, you actually can begin seeing them in the macro world. Now, this is more of an example than it is an exact story, but roll with it. Silk has a really cool sheen to it. Part of that comes because you've got the silk protein is forming the beta pleated sheets that are just repeated over and over and over again. Naturally curly hair has in the protein, the keratin protein, different R groups that cause different kinds of structural changes. You get alpha helical twists and things, and you generate curls. So we've got these different kinds of structures that you repeat it enough, and it actually changes the effect and look of the protein in the macro world. Now, the critical point here is the charge interaction and hydrogen bonding interaction. The bonds that are responsible for secondary structure are going to be things like polar, non-polar interactions. Let's see. Ionic bonds and charge interactions. And, of course, hydrogen bonds. And if we think about this in our bond strength chart, these are our distinctly weaker bonds, what is known in the official biz as them weak-ass bonds. Don't let the chemistry and the official terminology shake you, because it's just the way it's called. No, but it's a way for you to remember it. Them weak-ass bonds, they weak. Well, what that means is secondary structure is not all that tough. And you can begin jacking with the secondary structure. And, you can't touch the primary structure, but secondary, oh yeah, we can start screwing that up. Now, let's finish up here with the tertiary structure. The tertiary structure is defined as not neighboring R group interactions, but non-neighboring. Kind of non-neighboring, or let's just say, for the heck of it, long-range R group interactions. And now, we're going to take this protein structure here and begin folding it up. So we got the alpha helix up here, and then the big structure nonpolars, and then we got the alpha helix over here. We got negative charge interaction going on. And we can fold a protein up into all kinds of cool structures. The bonds that hold these things together look a lot like that, but just for completeness, let's write them down. Polar, non-polar guys. So oil and water ain't gonna mix. We got charge interactions. We got lots of hydrogen bonds. Again, not to get all technical, but the weak-ass bonds. All right. What this means is, primary structure, you ain't touching. Secondary structure and tertiary structure, they're delicate. And what we mean is, if you've got stuff that is dependent on pH, I'm just looking at the time here. These acid parts of R groups, the R groups that are acids, they are weak acids and they are weak bases. And the thing we know about weak acids and weak bases, if you take a weak acid, it'll act as an acid. That is, it'll have its hydrogen ion removed, its proton removed, at reasonably high pHs. But if you take a weak acid and throw it into a low pH, stronger acid solution, it doesn't act like an acid anymore. In fact, it'll begin to act like a base and actually change charge. And the same thing with bases. Weak bases are acting like bases. They've pulled in that OH group and they now are carrying a positive charge. They act like bases in lower pH solutions. You raise the pH up, and they now no longer have their charge on them. They can even become opposite charges. So changing the pH changes the charge of a bunch of different R groups. If we add salt, increasing the ionic concentration of a solution can cause different charged parts to either lay out or squish in tighter. The nonpolar parts start to freak out because they can't handle being in a real salty solution. We can begin throwing things off that way. Another great way to jack with secondary and tertiary structure is to play with the hydrogen bonds, and the best way you play with hydrogen bonds is to show them a little bit of heat. You raise the temperature up, and the hydrogen bonds begin to break. And certainly if you boil it, that's it, man, there's no hydrogen bonds hanging on. You lower the temperature, and things that probably should be kind of hanging around free, they might be able to get close enough that they hydrogen bond with somebody they shouldn't be. So temperature is a great way to jack with secondary and tertiary structure. And what we have stumbled into here is, in fact, the fundamentals of evolution, genetics, life, and it is this. This is the next thing you should tattoo onto your forehead. First, protein function depends on protein structure. And protein structure depends on amino acid sequence. And amino acid sequence is determined by DNA sequence. Consequently, you change a single nucleotide in your DNA, and you can potentially change an amino acid sequence. You change an amino acid sequence, and you can potentially change a protein's structure. You change the protein structure, you can change a protein's function. That is the basis behind evolution. As nucleotide sequence changes, we end up potentially changing an amino acid sequence that can potentially change the shape of the protein. So, we have hit on a number of really critical bits here, which we'll pick up on Friday. But this is the shiz. This is what drives the whole game. And that is protein structures and the fact that, sadly, they're held together by weak-ass bonds. If only protein structure were held together by covalent tough-ass bonds, we could probably live on Venus. We could live in all kinds of different really harsh environments. But no, we are delicate creatures because our proteins are so delicate. All right, that's all I have to say. Ciao.